Can you hear me? Yes, great, thank you. Good morning, everyone. So we'll get cracking on the neurofeedback bench to bedside. So the first speaker is Michelle Ramotte, and she's going to talk about the untapped potential of neurofeedback for basic science research. We will have questions at the end, so please use your app to ask questions. And I guess if you don't manage to use your app, you can also just raise your hand and we'll try to get to you. So without further ado, Michelle.
Thank you. Can you hear me? Okay, good, thanks. So first, thank you all for coming. I don't know I would have made it, so thank you. And I'm going to talk about the untapped potential of neurofeedback for basic science research. And I say that because this is the main question we have in neuroscience, right, in general. So what is this link between brain and behavior? That's what we're trying to ask. That's what basically all of neuroscience is trying to ask. And of course, it's a bidirectional link. And in most experimental paradigms, and this is certainly true for human imaging, but it's actually also true for animal experiments as well, what we usually do to ask how this link comes about is we manipulate behavior in some way, and then we see how that affects the brain. Or maybe we manipulate external stimuli in some way, and we see how that affects the brain, and maybe how that then in turn affects behavior, right? But that's not the real question we want to answer. The real question we want to answer is how does the brain cause behavior, right? How a particular brain state maps onto a particular behavior. That's what we would really want to know. And to ask that question, we need to know about causality. We need to be able to directly manipulate the brain, perturb it in some way, and see how that perturbation affects behavior. Now, there are very few ways of doing this, especially in humans. And I want to suggest that one such way is neurofeedback. And before I explain how we can do that with neurofeedback, I'll talk about two main approaches to neurofeedback, right? The first is explicit or task-based neurofeedback. And the second is implicit or covert neurofeedback. And this is basically, you can say, is the difference between direct control over the signal, so teaching people to directly control a particular signal that's coming from the brain, versus a more operant conditioning view of neurofeedback. So you have a particular brain state, and then you reinforce it in real time. But you're not necessarily teaching people to control it. Okay, this is the main distinction I want to make. Okay, so when you design a neurofeedback study, you have two important design decisions you have to make. The first is what feedback you're going to give. And the second is what instructions you're going to give to participants. Okay, so when I prepared this presentation, I didn't know I would be the first talk, so I was kind of hoping there would be a primer to neurofeedback before this, and there isn't, so I'll just stop briefly and do a brief primer to neurofeedback in case any of you don't know neurofeedback. In neurofeedback, what usually happens in fMRI neurofeedback is you look at the brain in real time while you're scanning people in the fMRI, and then you do one of two things, which is what I said before. You either translate that signal into something that people can perceive. So for instance, you want to teach them to control signal in the amygdala, right? So you look at activity in the amygdala, and you present the readout from that signal as a visual signal or something like that. So it might be a thermometer, and you want people to increase the strength of the signal, so you tell them make the thermometer go up or make the ball go up or something like that, right? So that's one approach to neurofeedback, where you're showing them the signal directly, you're translating it, and you're teaching them to control it, okay? But the other thing you can do is this more operant conditioning type thing. So you're not necessarily showing them the signal from the brain, you're just rewarding them whenever the signal is doing whatever you want it to do. But it doesn't have to be a direct translation, okay? Okay, so hopefully this will become a little bit clearer later on. So, okay, when you have a brain, you're interested in a particular brain state, okay? So for instance, say you're interested in face memory, or faces in general, okay? And you have this feedback signal that you can give to participants. And this feedback can be on this scale. We can imagine it on a scale from completely unrelated to whatever it is you are training to designed to optimally activate whatever it is you are training. So if you're thinking about faces, for instance, then a completely unrelated feedback would be maybe like sounds, right? Like an abstract sound, or maybe like an abstract visual image, right? Whereas feedback that's designed to optimally activate this region that you're targeting would be showing them faces. Okay? Is that clear? Okay. The other thing is what instructions you give to participants. So say, now again, you're interested in faces. And the instructions you give to participants can put them in the mental state that you can also imagine along this spectrum, from completely spontaneous, unrelated to the network that you're training, to designed to optimally activate this network that you're training. Okay? So completely spontaneous, you might not give them any instructions. Or you might just say, just try whatever it is, just try to do whatever it is that makes you get feedback. Whereas if you're trying to get people to optimally activate the network, you might tell them, imagine faces. Okay, you would give them an instruction that's supposed to activate this network. So if we go back to this link that I said we're trying to understand between brain and behavior, and now we're really trying to understand this link, like from the brain to behavior, you want to see how changing the brain might change behavior, right? So to do that, you really want to bypass these other two pathways. You don't want to change behavior, and you don't want to change external stimuli. So if that's what you want to do, for causality research, what you really want to concentrate on is having both the feedback and the instructions be as orthogonal as possible to the network that you're training, okay? Because that will let you bypass these other two pathways. And then you'll have a much more direct access to the brain. Okay. So the other, I call this beyond mapping as well. It was on the title, both because of where we are, but also because for a long time, neuroscience, both in humans but also in animals, has concentrated on mapping the brain. So we know this is the region involved in visual, we know this is an auditory region, we know this is involved in faces, and so on. And that's been hugely important, but we also know that there are networks. And the networks are important, and it's not just the regions, but also the interactions between them that are important for behavior. So, we really want to, I think this is the next step for neuroscience, is really starting to ask questions about networks and not just about regions. The other thing we know is that there are huge individual differences in just about everything. Okay, so people are very different in how they perform tasks. Even within a clinical population, you have differences in clinical symptoms. You definitely have differences between clinical and non-clinical populations, but there are huge individual differences. And there have been lots of studies, and I know this has been in the news lately, but there is a lot of evidence that correlations in spontaneous activity between different brain regions predict individual differences in task performance, clinical symptoms, like a whole bunch of things. So we know spontaneous activity is important, and we know that individual differences can be found in these networks of spontaneous activity. So what we really want to be able to do is ask what this spontaneous activity means, because that's really unclear. So it predicts behavior in some way, but it's very unclear what would happen if you change this spontaneous activity. Okay, so if we think about neurofeedback as a kind of operant conditioning, then, thank you, then we can use it to reinforce target brain states in real time across large-scale networks. Because the thing about this kind of neurofeedback is that you can really feedback anything that you can measure. Because what's really happening is you're looking at the brain in real time, you've defined the particular brain state that interests you, and whenever that brain state occurs, you give a reward. And because it's a kind of operant conditioning type of thing, you don't need them to know what the network represents, you don't need them to practice anything, you don't need to give them a strategy. In fact, you probably shouldn't give them a strategy, right? So it's very flexible in what you can do, and you can also use it on networks. So in this view, neurofeedback is not a conscious training paradigm, but it's more a neural perturbation technique, right? You're reinforcing particular brain states, you change them, and after you change them, you can ask what that did to behavior. And this is really good for studying spontaneous activity, because what you're really strengthening, what you're really feedbacking, is spontaneous activity, in a sense, right? Because you're not activating the network through either the feedback or the instructions you're giving to participants. So in a sense, what you're working with is spontaneous activity. So this is perfect for asking questions about spontaneous activity and the causality of spontaneous activity and what happens if you change it, which is really almost impossible in any other way. Okay, so I also want to add one more thing, which is also relevant for people who are interested in clinical neurofeedback. The main reason to do this kind of covert neurofeedback, as far as I'm concerned, is if you're interested in studying causality and basic science and spontaneous activity. But I think it could also be useful for clinical neuroscience, because if you think about the networks we have, like the way everything is set up, it's kind of the filters through which we experience the world. So if you have a network which through development or whatever is wired in a suboptimal way, and so for instance, say you are like me and you're terrible at remembering faces, then whenever you see a face, we assume that your networks for face memory are activating in a suboptimal way. And every time you see faces, you're reactivating this suboptimal network. And showing you more and more faces isn't going to change that, because I know I have seen plenty of faces, and it still hasn't improved my face memory, right? So it's not about just reactivating this network. You really want to change the network. So if you're just repeating and you're just presenting, you're reactivating this network, you might not be able to change it as much. Whereas if you bypass it, right, and I'm going to show you a cute dog. So say you were injured, and now you have, you need to go to physiotherapy, right? Because maybe your muscles are now kind of wired wrong, like since the injury, maybe you're limping a bit or whatever. And if you just continue walking, it's not going to change. But if you go to physiotherapy, they might give you exercises that could kind of push you back into the optimal way of walking, okay? So this is kind of the way I think of neurofeedback. So I think this covert neurofeedback might bypass these suboptimal networks in a way and allow you more plasticity than the more explicit type of neurofeedback. Okay, so before you believe me that neurofeedback is good, I need to answer a few questions. So the first is, can this kind of implicit feedback that I've been talking about, can it even work with real-time fMRI? Right, this is a big question. We know this kind of implicit feedback can work in animals with electrophysiology, right? This has been done before. You can train a monkey to increase firing rate in a particular neuron or change oscillations in different frequencies. So we know it can be done in principle. So, and all feedback in animals is kind of implicit. But we still don't know we can do it with real-time fMRI. Can we change networks? So I said this is great for changing networks, but can we change networks? Will this change the networks cause a change to the associated behavior? So I'm just going to go very quickly over 3 studies that hopefully answer these questions, and then we'll leave time for questions at the end. Okay, so this is a study I did a while ago, which the whole point of the study was to answer the first question. So can this kind of implicit feedback even work? So what we did is we brought people into the scanner, and they came in for 5 days, and we told them this is a study about reward. And you're going to hear good sounds. Thank you. And bad sounds. Excellent. And this is better than the laptop. And the sounds are inherently good or bad, right? So you know when you hear the good sound, it's a good sound. You know the bad sound, it's a bad sound. But it's also coupled to a monetary reward. So whenever you hear the good sound, you're winning money. Whenever you hear the bad sound, you're losing money. They know this. And their task is simply to press a button. You press one button whenever you hear the good sound. You press a different button whenever you hear the bad sound. And as far as they're concerned, that's all that's happening. So they think the sounds are completely random. Right? And they're not trying to control them in any way because they're sure that they're random. But what's really happening is we're looking at 2 different brain areas. So one is the FFA, the fusiform face area. The other is the PPA, the paracampal place area. These were chosen just because they're well-studied regions. They have nothing to do with anything. And we divided subjects into 2 groups, the FFA good group and the PPA good group. People in the FFA good group received good feedback or good sounds whenever activity in FFA was higher than activity in PPA. And they received bad feedback when it was the other way around. And of course, for the PPA good group, it was opposite. Okay? And the question is, so they're not trying to control the feedback, but they are getting the feedback. So they have an intrinsic wish to get more good feedback. Right? So the question is, will the brain learn to produce the activity that gives it good feedback on its own, completely implicitly? So what I'm showing here is how the brain changed over the course of training. So these are voxels that are significantly more correlated to the good region, be it FFA or PPA, right, depending on the group, on the first day during neurofeedback. And you can see there's nothing very much going on. But then by the second day and the third day, we already have these significant changes. So now there are many more areas in the brain that are more connected to the good region than the bad region. And if you look at what these places are, then you get regions associated with reward, right? So you get striatum and like a lot of reward regions, and also default mode network. Okay, so this was very nice. This was promising. It made us think that maybe this kind of implicit feedback learning can work with real-time fMRI. But we still have to answer these 2 questions. So can we change networks, and can we change, and will changing networks cause a change in behavior? Okay. So the second question is actually 2 questions. So can we change networks, and can we change them even if they're aberrant? Even if there's something about their connectivity which is aberrant in some way, through development, through whatever. And we decided to go to autism because there's a lot of work in autism about networks that are aberrant. So, and we also had a large data set in the lab. of people with ASD and how their networks differed from people without ASD. So we chose, based on this data, we chose 2 target regions who are most underconnected in autism compared with controls. And the degree of this underconnectivity is also correlated with symptom severity. So the more underconnected they are, the worse the symptoms. So these are these 2 regions over here, which I can't point to, but you're going to have to see. Like here, target 1 is in STS, target 2 is in somatosensory cortex. So this was a data-driven analysis. We just chose the regions that were the most underconnected. And then we also chose a control region which was slightly overconnected in our subjects. So in the ASD subjects that we had, it was slightly more overconnected to the other 2 targets than it was in the TD population. Okay? Okay, so now we're trying to train this little network. We want people to increase, so we're training high-functioning ASD subjects, 15 to 25-year-olds. Okay, and we're trying to get them to increase the connectivity between the 2 targets while at the same time decreasing connectivity between the targets and controls. So again, we bring them to the scanner, and now we do something slightly different. We look at the signal from these 3 regions at each time point, each TR, okay? And we ask a simple question. Over the course of this one TR, this previous TR, did the signal go up or down in each of the 3 regions separately, okay? And we're looking for this kind of pattern, where you either have the 2 targets going up together and the control goes down, or they go down together and the control goes up. I don't care which way they're going, as long as they're moving together and opposite from control. And whenever this happens, so they're in the right configuration, then I give them feedback. Okay, and this is an auditory visual feedback. I'll show you what it looks like in a second. And this completes the loop. Okay, so this is what the feedback looks like. There's supposed to be a sound as well. It also goes like da-dum, like the previous one. So we're going to try it again. No? Never mind. Okay, so you see they start with a blank screen. Can we just go back to the screen? I can go back. Okay. They start with this blank screen, and we tell them there's a picture hidden underneath the screen. And your job is to try and reveal the picture. And they say, what? What? He's like, no, just try and reveal the picture. And then what happens is they see pieces of the picture come up, right? Like they did. Never mind. They see pieces of the picture come up, and they hear like a good reward sound. And in the end, they get their score, which is how many squares they revealed. And then when they try, we do a few sessions, so in the next sessions, they can try and beat their previous high score, and they get a bonus. They get very into this. They get very excited. We call this the puzzle task. And they come up with all kinds of strategies, none of which have anything to do with the correlation between STS and somatosensory. Ah, there we go. It worked. Isn't it rewarding? Okay. So what we're looking for is improvement in the sense of like the changes in correlations in the way we were training and long-term changes. And remember, this is network that we're training, and we see, okay, that's exactly what we see. So we see an increase in correlation between the 2 targets over time. They came in for 4 days over 2 weeks, and then we brought them back for a follow-up, which was between a month and a year after the initial training. And we see that the changes we see are largely maintained in the follow-up as well. And we also see the opposite effect, which we trained, which was a decrease in correlations between the targets and control, which is also largely maintained in the follow-up. You see a little floor effect here as well. Okay, so we can change network, and implicit feedback works, but we don't know much about behavior yet because it's really difficult to do behavior in ASD, and especially this kind of change. We do have some preliminary data, but it's not super convincing because it's not the right paradigm to ask about behavioral change. So we thought, okay, if we want to go back and ask about what changes in behavior when we change networks, we should do something where we have a good behavioral measure. So we went back to fMRI. And this is from a different paper where we first located the networks that are involved in fMRI. And we said, okay, so we'll train the networks that are most predictive of your fMRI abilities. And we'll we'll take people who are terrible at fMRI, like me. And we'll see if we strengthen the networks that were weak in these people, will we see a change in behavior? So we started the study just before COVID, and we got 10 participants. And so I don't have enough data really to be sure. We saw a similar kind of change in networks, but now I want to show you what happened to behavior. So this is 10 participants, okay? And pre-training, you can see they're terrible. They're at about 2 standard deviations below the norm. And then in the follow-ups we do a week and 3 months after training, they're doing much, much better. So this is an increase of more than 1 standard deviation. And this is using the Cambridge face memory test. There's also the Cambridge car memory test. So we did the Cambridge car memory test also at the same time points. And there we see no change in the performance. So this is really promising. It's very nice. It's missing a lot of controls. It's good for grants and not much else. But we are redoing the study now with a slightly different approach, and hopefully we'll have more data sometime soon, and we can replicate this. Okay, so just to wrap up, what we do in my lab, and I think is a really nice approach for using neurofeedback for basic science, is first you want to characterize behavior and individual differences, right? So you want to have a behavior that is variable across, hopefully, the TD population. Then you can use the behavioral variance to find the neural variance that explains it, and you can form an hypothesis in spontaneous activity, right? And you can form an hypothesis about a neural network that might support it. So this is basically the first two steps are trying to find a network that you are interested in, that you have a clear correlation to behavior. And then you can directly perturb the network using neurofeedback and see how that changes behavior. Okay, and this gives you like a nice little causality loop. Okay, and just one final point about the neurofeedback. Again, for clinical research, I think this is also very useful for clinical because in a lot of clinical conditions, we don't actually know, we don't have a good sense of what the networks underlying the symptoms are. So I think using this kind of approach, which really asks about causality, might be good even when doing clinical neurofeedback, to first identify the networks that are most associated with behavior and have the biggest effect on behavior, and then using neurofeedback to change them. Okay, so thank you very much. This is my wonderful lab at Weizmann, and talk to us if you want to join us. Thank you.
Thank you, Michelle. So you can use the app to ask questions. We already have some questions, so I'll start with the first question we have. So how do you deal with participants doing strategies you do not want to have, like holding breath movements in an implicit neurofeedback paradigm? It's a very good question.
Yeah, so that is a very good question. We do tell people, so for instance, in the puzzle task I showed, people ask, like, what do you want me to do? And we generally say, just try, but just know that it has nothing to do with the way you breathe. So you have to stay still, and you can't change your breathing. So we do tell them explicitly not to do that. I mean, we can't control if they really do that or not. We do measure breathing while we scan, and we looked, like here, there was no correlation with head motion or breathing, so it doesn't seem like that's what they were doing. It's also not what they report doing, so we asked them at the end, like, what their strategies were, and they have a lot of strategies that involve eye movements, but not all participants have strategies that involve eye movements. They have very different strategies. So some of them said you have to think happy thoughts, or they have all kinds of strategies. So I think that doesn't explain what's happening, but it's true that they could have some kind of underlying strategy that we don't know about.
Okay, thank you. There's more questions coming. So people want to know if you're using a specific software or in-house scripts to compute your neurofeedback?
So the algorithm is an in-house script it's a very simple algorithm. It's also published, but that we use an in-house script for. We use AFNI, the real-time plugin, for doing all the real-time stuff. So AFNI real-time allows you access to the data, and then you can pull it and do whatever you want with it.
Okay. So is there a risk that changing network activity will have unforeseen negative consequences on cognition or behavior, and how do you monitor these side effects?
So, yeah, that's a good question. The only thing so there are a few things I can say to this. One is that this is still much less invasive than TMS, for instance. And we only do very short training sessions. So training sessions are about half an hour, like the actual training session, you usually no more than half an hour. We do like 4, 5, 6 of these. So I think the reason we're getting any change at all, like the fact that it works is astonishing. I think the reason we get any kind of change is because the changes we're making are somehow adaptive. And if you think about the network as being in a suboptimal state and you're kind of pushing it into a slightly more optimal state, then it could then be reinforced in real life as well, outside the scanner. So we actually had a control group for the ASD study, which I didn't talk about, where we tried, so instead we had the 2 targets in the control, and we swapped them, switched them. So now, like, these were the two targets and this was the control, and this is a network configuration which shouldn't happen, and we actually weren't able to modify it. So I think we need to trust the brain's robustness in a sense. Like, we're making very small perturbations.
Okay, so the next question, are there participants who cannot perform the neurofeedback task successfully, and if so, how many? What's the proportion?
Yeah, so in the ASD study, we had 17 subjects, and 15 of them succeeded, so we had 2 of 17. I think that's actually a very high rate. In most neurofeedback or BCI studies, it's more like 20-30% that are non-responsive, they call them. There's also this term that I found, which I love, which is BCI illiterate. It's like they just never can learn to control the BCI. So there's definitely a proportion, but because we got such a high percentage, I think it also depends on motivation and maybe the particular paradigm. So I think probably everyone could learn. Maybe some people take longer to learn, or they need like more guidance.
Mm-hmm. So I'll actually ask a follow-up question to that. So are you looking at the source of variability in neurofeedback response in your studies?
Yeah, so that's something we're very interested in. It's hard because it's hard to get a large cohort for a neurofeedback study because it's so involved and each person has to be scanned so many hours. We did look at the reward response as a possible source of variance, like people who were more rewarded. But it seems like there might be something there, but it's hard to tell because it's a small cohort.
Okay, we still have some time, so people want to know what kind of control conditions you're using for your neurofeedback experiments. You gave a couple examples. I don't know if you want to give a few more.
Yeah, so what we did for the ASD study was have, like I said, like the different configuration of the network, which basically gives you almost sham neurofeedback in the sense that it's orthogonal. Like, you get completely different feedback than you would have gotten for the original network position. So it's a kind of sham, but it's still coming from your own brain. Like, it's not sham in the sense that it's just random feedback that has nothing to do with your own brain activity, which I think is better. I think the best kind of control would be to have like a double dissociation. So instead of training people on sham, like kind of like we did for the first study I showed, where you had people in the FFA good or the PPA good group, so you have an internal control for that. So you're training people in one direction and training in another group in the other direction. Or if you have two different tasks, where you have two different predictions on what the behavioral change would be and what the network changes would be, and you train one group on one particular network and the other group on the other network, I think that's the ideal control. It's just, it takes a lot of time.
Okay, I'm going to select one last question. So this one is a technical one about the TR. So did you do any tests to increase the duration to more than one TR? Is there an optimal time window? I think this is a good technical question.
Yeah, so I actually have a paper about this, so it's an excellent technical question. So before choosing this, I actually did, okay, you can collect data and then run it offline. It's not going to be neurofeedback data because you don't have the feedback element in it, but you can look at the signals themselves and just kind of try and simulate what would happen if you did different TRs or different, if you used like 3 TRs or 4 TRs, if you had 4 regions or 5 regions or 3 regions, right? And you can just see how successful that is just from a SNR point of view, right? So the reason we chose using just 1 TR and having just the 3 regions was because that gave us the best SNR. So the real-time signal is very noisy, and you want to be able to do something that's still correlated to the good, clean signal afterwards. So we did a bunch of simulations, I did a bunch of simulations, to try and find like what the optimal configuration would be, and that was it. So.
Great. Let's thank Michelle again for being here.
Thank you, and thank you again for coming.
And thank you all for your questions. So I'll invite Michelle to answer these questions on the app. I think it's possible, maybe. Yeah, don't worry. You'll have the whole conference to manage to answer them. There's quite a few still. And our next speaker, unfortunately, is not able to be here, but we have her video. So the next speaker is Professor Michelle Hampson, and she'll be talking about, hopefully there'll be a video soon, but she'll be talking about potential applications of neurofeedback to clinical studies, and she'll explain also how...
Hello, my name is Michelle Hampson, and I'm going to be talking about the time course of clinical...
And so she'll be talking about how about the time course of potential clinical changes in after neurofeedback interventions in different clinical populations. So I hope that will work. And Here's Michelle.
Hello, my name is Michelle Hampson, and I'm going to be talking about the time course of clinical change we found in our neurofeedback clinical trials. So when I say neurofeedback clinical trials, I'm referring specifically to fMRI neurofeedback. As you've heard from Michal, this is a technique that has a lot of potential from a basic science perspective because it allows us to change very targeted aspects of brain function and see how that translates into changes in mental function. But it's also a technique that has a lot of clinical potential, and that's what our group is particularly interested in. So if you can train people to control symptom-relevant brain function, and that training, that the effects of that training actually persist, then this could be potentially a form of treatment. But a key issue there is whether those effects persist, because you can't really bring people in every day or two to train them, to keep refreshing the training their whole life. So we need there to be some persistence in the effects induced. And there is some evidence from the existing literature that this is feasible for many applications. Some of the earliest fMRI neurofeedback studies followed subjects up for a couple weeks and reported that the effects were still there a couple weeks later. More recently, studies have followed subjects for longer, months to even years, and reported that effects were still persisting. And this is consistent with the EEG neurofeedback literature, which has also reported that some of the effects can persist for months to years. So at least for some applications, in some instances, it seems like you can induce very persistent effects. But for any application, it's actually useful clinically to understand how the effects unfold over time, so that you can see when they wax or wane and when it might be a good time to give a booster, for example. So we're very interested in just mapping out the pattern of clinical change associated with our intervention. Now I'm showing here schematic diagrams. These are not real data, but they're showing on the x-axis time, the neurofeedback intervention occurs during this blue period, and then we're following subjects up over the weeks following. And on the y-axis, we're showing the symptoms in the real and the sham groups. A decrease in symptoms is, of course, an improvement. So if your intervention decreases symptoms in the real neurofeedback group during training, that's good. But if it regresses back to baseline pretty quickly, that's a transient effect, which is not going to be super clinically useful. So what we're actually hoping to see in our subjects is that the neurofeedback will improve their symptoms and that they'll stay in that improved state in a persistent way for a while afterwards. But of course, the reality is we could see any kind of complicated pattern of change, and we don't know what that's going to be, but we want to look at that and map it out. So I'm going to describe to you what we found on this front in two different clinical trials we ran. The first of these was a study of obsessive compulsive disorder run by Mariella Rance, where we were targeting a ventral frontal region and trying to improve their OCD symptoms as measured with the Y-BOX, which stands for Yale-Brown Obsessive Compulsive Symptom Scale. The other study is a completely different neurofeedback clinical trial. We were recruiting adolescents with Tourette syndrome. The study was run by Chris Walsh. The kids were being trained to control activity in their supplementary motor area with the aim of reducing their tic symptoms as measured by the YGTSS, or Yale Global Tic Symptom Severity Scale. So we're talking here about two completely different neurofeedback clinical trials, recruiting different patient populations, targeting different aspects of brain function, trying to change different aspects of different clinical symptoms and using different clinical scales to assess the effects. But both of these studies showed a kind of interesting pattern that seemed to be common across them. So that's what we're going to be talking about today. I'm going to start by describing the OCD study where we first noticed this pattern. This is a randomized double-blind clinical trial. So subjects were recruited and they're assigned to either receive real neurofeedback or a sham form of neurofeedback. And we assessed their symptoms at 4 points in time before the training. What we call post-neurofeedback, which is typically about 3 days after the training, 2 weeks after, and 1 month after. And to give you a sense of what the training was like in this study, I've got a little video that shows you what one of our subjects actually saw during the training. The white arrow pointing forward is cueing them to rest and relax and look at the neutral image. But at other periods of time, there will be a red arrow pointing up, cueing them to increase activity in their target brain area, or a blue arrow pointing down, cueing them to decrease activity in the target area. And during those periods, a contamination-related image will be shown. So this subject is just resting and looking at the activity in their target region, as it's shown in this line graph at the bottom of the screen. now it's going to turn into an increased block. So their goal now is to get the line to go up. And they can look at this contamination image of a dirty stove and think, oh, that's a food surface, it really should be clean. Whatever's going to be helpful for activating this anxiety-related brain area. And now it's a decreased block, so their goal is to get the line to go down. They're expected to look at the provocative image, but they can reappraise it, whatever's helpful. So they basically get lots and lots of practice trying to get the line to go up in the red blocks and come down in the blue blocks. And they can see how successful they are. This subject was quite successful. And we felt that perception of success would impact motivation because it's easier to stay engaged if you feel like you can do this somewhat. We also felt that perception of success would impact placebo effects because they know they're trying to control a symptom-relevant brain area. If they can see they're good at it, they're more likely to have a large placebo effect. So because we felt that controlling for perception of success was very important, we used what's called a yoke sham control. Yoke sham subjects are matched one-on-one to real subjects, and they see exactly the same time courses as the real subjects and are misled to believe they're getting information from their own brain, but they're not. They're seeing exactly what their matched neurofeedback subjects saw. To the extent that that person managed to get the line higher in the red than the blue blocks, they will be misled to believe they're having the same level of success. So that's all I'm going to say about the methods of this study. If you want to know any more, we have a methods paper with a cheesy video you can watch. In terms of the pattern of clinical change we found in this data, we did see that the real neurofeedback subjects showed symptom improvement that exceeded what we had in the sham group during training, which was encouraging. Then in the 2 weeks following training, both groups showed some symptom improvement. We expected at the month follow-up that they would both start regressing back to baseline as placebo effects wore off, and the sham group did, but the real neurofeedback group continued to show symptom improvements. So this was actually quite surprising to us. Just to reiterate what we've been thinking, we were hoping to rule out a transient effect where they had symptom improvements and then regressed back to baseline. We were hoping to see a stable effect, but what we actually saw was even better than that. We saw that symptoms continued to get better for the weeks that we followed them up. And in fact, the biggest difference between the two groups was at the month follow-up, not immediately post-training. So this might be something idiosyncratic to OCD, but we happened also to have this other data set of Tourette syndrome data, and we thought, well, let's just look at this data and see if we see a similar pattern. So this study, unfortunately, was not a randomized design because for power reasons, we decided to use a crossover design where subjects are recruited and randomized to either receive real and then sham or sham and then real. So every subject who came in for this study had to have 7 consecutive scans, an initial assessment scan, then the 2 scans associated with the training of the first leg of the intervention, then a midpoint assessment scan, 2 more training scans, and then a final assessment scan. So as you can imagine, this is a very logistically challenging study to run. This was run by Chris Walsh, who did a great job collecting the data, but about I think the second subject into this study, he had just finished running the first leg of the intervention, and the snowstorm Nemo hit the Northeast, and the entire MR center closed down. And I get this panicked email from Chris that he can't do the second leg of the intervention in a timely way because the MR center is not open. And I said, don't worry, that's not a big deal. You can just pause the study for a while, and then before you resume with the second leg, tack on an initial assessment scan for that second leg so that we have a pre-assessment. So once he realized that this was an acceptable design to use for subjects, he ended up using it to get around a lot of other scheduling difficulties. So in the end, about half of our subjects had this design, and about half of them had this broken apart design where we had 2 separate legs of the intervention. So this, differences across subjects is not typically ideal in a clinical trial, but this actually turned out to be a feature for us because these subjects where the intervention was broken apart, that allowed us to better map out how symptoms were changing over time in these subjects after the first leg of the intervention, and I'll come back to that in a second. So when we got the finding we had in the OCD study, where symptoms continued to improve over weeks after the training, I thought, well, what would this mean for the Tourette syndrome study? So if we had a subject come in and get real neurofeedback, and then their symptoms were continuing to improve not only during the neurofeedback, but during the weeks afterwards, well, that improvement that occurred during the weeks afterwards would be occurring during the sham sessions. And so it would be attributed to sham. So this is terrible for our power. The second leg of our intervention is going to be, terrible data because our control intervention is going to look much, much more effective than it actually is. And what's actually caused by the real intervention is going to be attributed to the control intervention. So how would that look in terms of the two different legs of the intervention. Well, what we would expect to see is nice clean data from the first leg because there's nothing contaminating it. And then a second leg where the data are much less nice, and the sham subjects in the sham intervention in particular showing much greater symptom improvement because it was actually having symptom improvement driven by the preceding real. So we decided to break the data apart and look at it in that way. When we looked at the symptom changes in the real and sham in the first leg, as expected, they looked beautiful with much better symptom improvement in the real than the sham. But when we looked at the second leg, indeed, it looked nowhere near as good. And the changes in symptoms in the second leg of sham were more than twice the improvements seen in the first leg of sham. So it really did seem like there was possibly a similar effect to what we'd seen in the OCD study, where the prior leg of real was bleeding into the second leg of sham and causing symptom improvements during sham that were actually driven by the preceding real. So we decided to look at this a little more closely. We could look at the precise timing of this post-intervention assessment. So to get information about how the sham intervention affected symptoms over time. We could take advantage of the fact that scheduling was variable at the MR Center, and this assessment sometimes was very close after the sham intervention started in some subjects and quite a while after in others to map out the temporal changes a little bit. And furthermore, for the subjects who had the broken apart intervention, we can use the pre-assessment scan of the second leg of the intervention to give us even further out information about how their symptoms were changing over time after the intervention. So we do have some information that allows us to map out how symptoms are changing over time after the sham training. And similarly, we have the same kind of information for the real training, but we have even more information for the real training. If we assume that the sham doesn't really do anything, then we can actually take these post-sham assessments as very far out in time assessments of the original real intervention. So that gives us a lot of power for looking very far out in time. And then we can use the second leg of real as well because we don't expect it to be contaminated by the prior sham. So we actually have quite a lot of data allowing us to map out how symptoms are changing over time after the intervention for the real intervention. So when we throw all these data into a scatter plot, here red is the real intervention, blue data points are from the sham intervention. On the x-axis, I'm coding the number of days after the start of the intervention that assessment was collected, and the y-axis is coding change in symptoms from baseline at that assessment. And you can see that symptoms seem to be improving over time for literally weeks after training in following the neurofeedback intervention. In contrast, symptoms don't seem to be changing much over time in the sham. So this is very consistent with what we saw in the OCD study. And we decided that after doing these qualitative analyses that we should really do a statistical quantitative analysis to see if this effect is a significant effect that's shared by these two studies. So we recruited a statistician, Brian Pittman, to help us with this. And we wanted to roll together the data from the OCD study and the data from the Tourette syndrome study. These are, of course, two totally different data sets. You know, the measures are YBOX and YGTSS. These are different clinical scales that have different means and ranges, et cetera. So we really needed to normalize these data in order to combine them. And we did this in a couple of different ways. I'm going to describe just one of these, which we call our Z-score analysis here. So the way this worked is that in the OCD study, we had all these YBOX measures, some at baseline and some collected post-intervention. We took just the baseline measures from both groups, including both real and sham subjects, but only looking at baseline. We computed the mean YBOX score and the standard deviation of the YBOX scores at baseline. Then we normalized every single YBOX data point in the data set, subtracting the mean and dividing by the standard deviation. So we put them all into a Z-score space based on the baseline ranges that we saw. We did a similar kind of normalization of the Tourette syndrome data using the YGTSS scores. And then we had both of these data sets in a Z-score space so we could roll them together. And we did a mixed model analysis using fixed effects for our group. Latency was our time variable. This is the number of days from the start of the intervention that assessment was collected. For baseline assessments, it's coded as 0. And for all the others, it's number of days since the start of the intervention. What we're really interested in is a group by latency interaction, because this is what's going to tell us if the neurofeedback subjects were improving more over time than the sham subjects. So when we did this mixed model analysis, sure enough, we found a significant group by latency interaction. And when we explored the nature of that interaction with post-hoc analyses, we found it was driven by a significant symptom improvement in the real group and no significant change in the sham group. So that was really encouraging, that real neurofeedback in this pooled data seems to be doing more than the sham in terms of helping symptoms. But it doesn't per se indicate that symptoms were improving, strictly speaking, after the intervention had ended, because it's including the changes that occur during the training. So we wanted to really ask the question, are symptoms continuing to improve after training is over? So to get more specifically at that, we decided to throw out all the baseline data and rerun the analysis using only measures that were collected post-intervention. So now all of the data going into our analysis are collected post-intervention. If we get an improvement in symptoms over time in the real group, this is an improvement that occurred after training. So we ran that analysis, and once again, we got a significant group by latency interaction. And when we explored the nature of the group by latency interaction, it was again driven by significant symptom improvement in the real group, this time, strictly speaking, after the training was over, and no significant slope in the sham group. So this is evidence that the effects of neurofeedback were growing over time after the intervention had ended in this combined data set of OCD and TS data. So you might say, well, maybe this is something weird about one of those studies, and it's kind of driving things and just carrying the result. So we decided to also explore whether the study, TS or OCD, was driving this result. So we included a binary variable coding whether each data point came from the OCD study or the Tourette syndrome study in the model. We also included all the interaction terms of this variable, and none of them were significant, and it did not change the results. In particular, we still got a group by latency interaction in the combined data set. So this indicates that across these two clinical trials, neurofeedback was yielding symptom improvements that grew after the intervention, specifically the real intervention, more so than the sham. So this was quite surprising when we first found this result, and we analyzed the data in multiple different ways to make sure that this was not like some sort of artifact of the way that we normalized across these studies or anything. And I refer you to our paper if you want the details of that, but really we found this finding was very robust to a number of different analyses. And so after we found this, I happened to go to lunch with Nick Turk-Brown, who's another neurofeedback researcher at Yale, and I was telling him about this, and his first reaction was, my goodness, we had the same effect in an open label study of depression that we ran. We didn't really say much about it, but if you go and look at the paper, you can see the effect in the figure. Sure enough, I went and fished up that figure, and this is it from the paper. The follow-ups were collected at week intervals after the training, and here the subjects were improving in their depression symptoms for literally weeks following the neurofeedback training, exactly the same pattern that we were seeing in our studies. And then I started searching the literature to see, what other published studies had data that could be relevant to whether they were expressing this effect or not. And I found quite a number that seemed to suggest the same pattern. I'm not going to go into details about these, but a basic science study that showed just a huge increase in effect 3 to 5 months later. We have also resting state connectivity studies that seem to show similar patterns. I'm not saying that these continuing changes in brain and behavior after training are going to be there in every single fMRI neurofeedback study, but it is a pattern that seems to be present in a lot of different applications. And papers have come out since we published our paper, also reporting that they're getting this effect. So it's been replicated in other studies. So in short, this is a pattern you might see if you're going to run an fMRI neurofeedback study yourself, and you should design your paradigm accordingly. So what I most want to emphasize on that front is that you should follow up your studies, right? If you don't follow up your studies for weeks to months, you could be missing the time point of greatest effect and underpowering yourself. So for example, if you have a pattern like ours where the neurofeedback improves symptoms somewhat more in the real than the sham group, but it's not really clinically meaningful. You might say, well, this is too small. It's not going to be clinically useful, the effects of this intervention. But actually, if you followed these subjects for weeks, you might find a whopper of effect if your pattern looks like ours at the month time point, for example. And when you consider how much time and money and effort it takes to run a neurofeedback intervention, bring people in for multiple sessions of real-time fMRI neurofeedback, compared to how easy it is to phone them up and do a clinical assessment, a 20-minute assessment to gauge where their symptoms are at that time point. It's really well worthwhile to tack on a few clinical assessments later on in order to make sure that you're measuring the time point of greatest effect for your intervention. Also, I would advise against using crossover designs, live and learn. And third, I want to highlight that one of the challenges for people doing fMRI neurofeedback research is figuring out how many sessions of training to use. And a popular approach people often adopt is that they'll interleave assessment scans and feedback scans, and then they'll just see, when do symptoms stabilize, and they'll assume that all the feedback scans up to that point were helpful and were contributing. And then the subsequent ones where symptoms aren't continuing to improve aren't doing anything. But that reasoning is flawed. That reasoning is based on an assumption that the symptom improvements caused by a given feedback scan occur during the feedback scan, when really our data have shown pretty clearly that in many instances they occur those changes can occur after. So you could have a single feedback scan that causes symptoms to improve for a month. And if you use this approach to try to figure out how many feedback scans you need, you might conclude you need like 12 of them, right? When really only the first one was effective and was doing anything. It's just that its effects took a long time to unfold. So in short, if you use this approach to try to figure out how many feedback scans are optimal, you're at risk of grossly overestimating the optimal number. And that's going to cost you a huge amount of time and money later on when you're running 12 scans instead of 2, for example. So sadly, the only really dependable way to optimize number of sessions in a real-time fMRI neurofeedback study, in my opinion, is to run a multi-leg intervention. And that takes a huge amount of resources because you have to randomize people to multiple different groups where one group gets 1 session and is followed, another group gets 2 sessions and is followed, and so forth. And that's very resource intensive. I do appreciate that, but sadly, I think that's the implication of our finding. So I just want to acknowledge the contributions of people to this work. Mariella Rance ran the OCD study, and Chris Pittinger was our primary clinical collaborator on that study, and a lot of the OCD research clinic staff helped us with that. The Tourette syndrome study was run by Christopher Walsh. Our primary clinical collaborator was Dennis Subudolsky, and the Yale Child Study Center Tic Disorders Clinic helped us with that study. Dustin Chinos has helped a lot with some of our technical issues, and Brian Pittman is the statistician who helped us with the omnibus analysis. He wouldn't give me his picture, so I had to sketch him here. So this work has been supported almost entirely by NIMH, but we've had some foundation grants, which have also been super helpful. So thank you for your time.
Great. So obviously Michelle is not here, but you're able to use the app and ask questions, and I will let her know, and she'll answer them either on the app or some other platform that I'm not aware of yet. You can also email her if you have questions, if you want to talk to her more directly. So we'll move to the next speaker, which she's also unfortunately still en route. So we also have her video. So Zina will be talking about how to use neurofeedback in stroke patients, and will give an example of a randomized control trial that she ran recently. If we can find the video. Oh, great, fantastic.
Hello, my name is Zina Sanders, and I'm going to be presenting.
I'll be able to answer some of the questions at the end. Okay, so, sorry, could you repeat? Could we dim the light, please?
And I'm going to...
Is it possible to dim the light? Great, thank you. Much better, thanks. Great. So we'll start then. Thank you so much for your attention.
Are very common after stroke. It's been estimated that about 75% of stroke patients suffer motor impairments immediately after stroke. And while some proportion of these patients recover functioning, approximately between 5 and 20%, there is still a significant proportion of patients who are left with ongoing, long-lasting motor impairments. And these motor impairments can have a very negative impact on patients' abilities to perform activities of daily living, impacting patients' independence and their mobility, and is also related to decreased health-related quality of life. Rehabilitation can improve motor outcomes, and especially it has been argued that greater intensity and duration of training is related to greater recovery. But this often requires one-to-one sessions with a physiotherapist, which can be very time-intensive and expensive. Also, traditional rehabilitation is typically focused on the stroke-affected limb, but research has started to explore the possibility of targeting the brain to see whether this can help improve motor functioning or boost rehab effects. After a stroke, we can see that there is altered brain activity. So when a healthy participant moves one of their limbs, we get a very lateralized brain activity pattern in the contralateral motor cortex. In comparison, when a stroke patient moves their stroke-affected limb, we observe a much more bilateral activity pattern, with increased activity in the ipsilateral motor cortex. This more bilateral activity pattern has been associated with worse outcomes in some patients, particularly those with milder impairments, but it's also thought to be compensatory in other patients, particularly those with worse motor impairments and more damage to the corticospinal tract. Therefore, depending on the patient, it may be beneficial to either try to decrease this ipsilateral activity, normalizing the brain activity, or to increase ipsilateral activity in order to help them compensate. And there have been several brain stimulation studies that have attempted to target this bilateral activity pattern in the hope of improving rehab outcomes. An alternative way in which you could target brain activity in a more endogenously driven way is through the use of real-time fMRI neurofeedback. During real-time fMRI neurofeedback, you have a participant inside the scanner, and you pre-process and analyze their data in real time and extract a feature of interest, for example, the percent signal change in a specific region of interest, which you then show back to the participant inside the scanner using a visual display. You then ask them to try and modulate the visual display, which corresponds to them modulating their brain activity. To date, there seems to be some promising evidence that people are able to use fMRI neurofeedback to modulate brain activity. In this recent systematic review that looked at previous fMRI neurofeedback studies, they found that of approximately 100 previous studies that they looked at, 2/3 of the studies showed that people were able to modulate their brain activity using neurofeedback, and another approximately quarter of the studies showed that there were at least some responders and some non-responders. So it seems that in general, people seem to be able to use neurofeedback to regulate brain activity. There's also been some evidence of promising behavioral results. So not all studies include behavioral measures, but of those that did, we can see that there have been some promising behavioral effects on a variety of different domains, ranging from cognitive functions to emotions and physiological responses. So if we can use neurofeedback to modulate brain activity, and if this can have a positive effect on behavior, then this could be a potentially interesting therapeutic tool in disorders which are associated with abnormal brain activity. And people have tried to use fMRI neurofeedback in a variety of different clinical populations to varying success. But to date, there hasn't yet been any randomized trials in stroke patients. So here we wanted to carry out a randomized trial in chronic stroke patients, which was double-blind and sham-controlled. And we wanted to see whether the stroke patients could use the neurofeedback to increase lateralization of their brain activity. Our primary outcome measures were whether or not the patients could change the lateralization of their brain activity during neurofeedback, so measuring whether or not they could actually use the neurofeedback. And our second primary outcome measure was change in performance on the Jepsen-Taylor test, which is a motor assessment that I will describe later on. We also had a bunch of secondary outcome measures, which included whether or not there was any change on clinical measures of upper limb functioning. For this, we used the ARAT and the Fugl-Meyer, and also whether there was any change in the white matter tracks after neurofeedback training, seeing as there had recently been some work showing changes in the white matter after neurofeedback. So we recruited 24 chronic stroke patients who were more than 6 months post-stroke with mild to moderate motor impairments, and they were randomized into the real or the sham group based on time since stroke and their baseline score on the ARAT. So we ended up with 12 patients in each group, and here on the right you can see their baseline characteristics, and you can see that they're fairly well matched on these two factors that we randomized them on. So the patients were invited to attend 6 experimental sessions with us. So first they came in for a baseline session during which we did a baseline MRI where we acquired functional and structural data. And they also completed a bunch of motor assessments. Following this, they then came in for 3 neurofeedback training days, on which they also completed the Jepson-Taylor test on each day. And then they came back for 2 follow-up time points. So one week after the neurofeedback training, where we repeated all the same measures as they completed during the baseline session, and 1 month later, where we just completed the motor assessments again. So I'm just going to describe in a little bit more detail what the neurofeedback intervention involved. So like I said, the patients came in for 3 neurofeedback sessions, and they were usually on a Monday, Tuesday, and a Thursday. And they always first completed a functional localizer, in which they were instructed to open and close both of their hands. And then the region of interest for the neurofeedback training was placed over the peak activity in the motor cortex. Then during the 3 runs of neurofeedback training, they saw 2 bars on the screen, with the red bar reflecting activity in the stroke affected hemisphere, and the blue bar reflecting activity in the unaffected hemisphere. And during the neurofeedback blocks, they were instructed to make different movements with their stroke-affected hand to try and increase the size of the red bar while keeping the blue bar as low as possible. So this corresponded to them increasing activity in the stroke-affected hemisphere while keeping activity in the unaffected hemisphere as low as possible. Before and after the neurofeedback training, the participants also completed transfer runs where they just saw the instructions rest or move right or move left depending on which arm is stroke-affected. And during the movement blocks, they were instructed to use whatever strategies or movements they had found effective during the previous neurofeedback blocks. So this was to test whether they could maintain any changes achieved with the neurofeedback when the feedback is removed. The sham group had exactly the same tasks and the same instructions, but rather than seeing the bars reflect their own brain activity, they just saw a video of a previous participant from the real group. So first of all, I'm going to be going through the results from our primary outcome measures. And so in order to assess whether they could use the neurofeedback to increase lateralization of their brain activity, we're going to be looking at the data from the neurofeedback runs themselves. In order to assess laterality of brain activity during neurofeedback training, we calculated a laterality index based on the activity in the regions of interest used for neurofeedback training. This resulted in values between -1 and 1, with -1 indicating activity lateralized towards the unaffected hemisphere, and 1 indicating activity lateralized towards the affected hemisphere. So higher positive values would indicate that participants were able to use the neurofeedback to increase lateralization towards the stroke-affected hemisphere. So here I've plotted the results from the real and the sham group, and I've plotted the laterality index over the 3 runs of neurofeedback training on each of the 3 training days. Individual patient data is plotted here with the grey line, and as you can see, the individual patient data is very variable. So we first wanted to check whether or not there was any learning occurring within training days. And for this, we took the average of all of the first, second, and third neurofeedback runs, and then we ran a linear mixed model with group and day as fixed factors and baseline laterality index as a covariate. And when we did this, we found that there was a significant interaction, and this appeared to be driven by patients in the real group increasing the laterality of their brain activity between the first and the third neurofeedback run, whilst the sham group did not change their laterality index over the runs. We then also checked whether or not there was any learning over neurofeedback training days, and so for this we took the average of the laterality index over all the runs within the training days, and then ran another linear mixed model, this time with group and day as our fixed factors and baseline laterality index as a covariate, and this time there was no significant main effects or interactions. So there is some indication that the real group appears to be able to use the neurofeedback to increase laterality across the runs within the training days, but as you can see, the performance is pretty variable and there isn't any evidence of a carryover effect across the neurofeedback days. So then we also checked whether or not patients can maintain neurofeedback learning, so whether they can continue to modulate brain activity without feedback. And for this, we looked at the data from the transfer runs, which were before and after the training, in which the patients did not see any feedback. They were just asked to use the strategies that they found effective in the previous neurofeedback runs. So for the transfer runs, we extracted the percent signal change from the regions of interest used for the neurofeedback training, and again calculated a laterality index, which I plotted here for the real group and the sham group, before and after the neurofeedback training on each of the 3 training days. And when we carried out a linear mixed model on the change in laterality index on each day and group, there were, unsurprisingly, from when you look at these graphs, there was no significant main effects or interactions. So there wasn't any evidence of a transfer effect on a group level here, but as you can see, the performance was quite variable. And there was also quite a lot of variability in how well people could use the neurofeedback. So we also decided to have a look at whether this transfer variability is related to variability in how well people could use the neurofeedback. So we calculated the average transfer change as the average pre-post difference for each of the training days, regressing out the baseline laterality index. And we also calculated a neurofeedback success index, which was the average slope over runs within the training days. And when we correlated these two things together, we found a trending positive correlation for the real group, where patients who performed better on the neurofeedback, as assessed using the success index, also had a greater pre-post change in the laterality index during the transfer runs. And this was not the case in the sham group. However, this didn't quite reach significance. So our second primary outcome measure was change in performance on the Jepsen-Taylor test. The Jepsen-Taylor test is a timed assessment where patients are timed in how long it takes them to complete 6 different subtasks, ranging from things such as picking up small objects like paper clips and buttons, to picking up filled or empty cans. And what we were interested in is whether there was any decrease in the time taken to complete these tasks after training. So here I've plotted the time taken for the two groups across all of the subtasks of the Jepsen-Taylor test. And when we ran a linear mixed model on this data with group and day as fixed factors and baseline time as a covariate, we saw that there was a significant main effect of time, but there was no effect of group or group by time interaction. Therefore, patients were getting better over time and getting faster, irrespective of what group they were in. So in line with some previous studies, we also carried out an exploratory analysis where we divided these 6 subtasks up into tasks that measure more gross and more fine motor function. So when we did the same analysis, just looking at performance on the gross motor tasks, we saw that there was a significant main effect of group, with patients in the real group having faster performance on these tasks than patients in the sham group. In comparison, when we looked at performance on the fine motor tasks, there was no group difference between the real and the sham group, and the two groups performed quite similar to each other. So although our primary outcome measure was negative, there does seem to be some nice indication that there does seem to be a persistent improvement on gross motor function after this relatively short period of neurofeedback training. So next I'm going to be moving on to our secondary outcome measures. And these were all measures that were acquired during the baseline and at either one or both of the follow-up time points. So first off, for our clinical measures, we carried out the ARAT and the Fugl-Meyer. So in the ARAT, patients have to do a variety of different activities like pouring water into a cup or picking up marbles, and this is a measure of activity limitation. The Fugl-Meyer is more a measure of patients' motor impairments, so they are assessed on their ability to perform things like shoulder abduction or elbow extension. So here I plotted the patient's scores on the ARAT before the training and at the 2 follow-up time points. And although there was a main effect of time, so patients were getting better and scoring higher on this test over time, there was no group effect, so neurofeedback didn't seem to be boosting this improvement at all. And for the Fugl-Meyer, the data looks very similar, with patients getting better over time, but in this case, the main effect of time didn't quite reach significance, and there was also no significant main effect of group. So unfortunately, we weren't able to show any effect on the clinical measures of upper limb function. Finally, we were also interested in looking at white matter changes post-neurofeedback. This is something we had looked at previously in a study with healthy participants who were asked to bidirectionally change ipsilateral brain activity, emulating the 2 different activity patterns that could be targeted in stroke patients that I discussed before. So participants had to make movements with their left hand and had to try to either decrease ipsilateral activity in a condition called the dissociation condition, or to increase ipsilateral activity in the association condition. And we found that healthy participants could indeed bidirectionally alter activity in the ipsilateral motor cortex, but interestingly, we also found changes in the white matter of the corpus callosum that were directionally related to the condition that the participants were in. So FA in this area in the corpus callosum was increasing in the association condition and decreasing in the dissociation condition. So this provides some evidence that neurofeedback could be used to induce structural brain changes. In the current study, we were interested in looking at changes in FA asymmetry of the cortical spinal tract. This is a metric that has been associated with motor impairments in stroke patients. So FA asymmetry is a measure of the difference in FA between the two cortical spinal tracts. And usually higher FA is associated with worse motor functioning. And this is something we could also replicate in the baseline data of our current study. And if you're interested in seeing some more data on relationships between brain structure and impairment, come check out poster MT752. So we compared this FA asymmetry measure before and after the neurofeedback training in our two groups. And here I've plotted the change in FA asymmetry with lower numbers indicating a reduction in FA asymmetry, so the two tracks becoming more symmetrical. And what you can see is that the real group is reducing asymmetry, whereas the sham group seems to be slightly increasing asymmetry. And when we did an ANCOVA on this with baseline as a covariate, we saw that the real group had significantly lower FA asymmetry compared to the sham group at the follow-up. So then we also wanted to have a look at whether this effect is driven more by changes in one of the cortical spinal tracts over the other. So here I've also plotted change in FA in the two cortical spinal tracts separately, with the affected hemisphere on the top and the unaffected on the bottom. And when we did a group by hemisphere ANCOVA, unfortunately it didn't quite come out in the statistics, but there was some indication that this was probably more likely driven by change in the affected cortical spinal tract of the real group than by changes in the unaffected cortical spinal tract or in the sham group. But like I said, unfortunately this wasn't significant. But what was interesting is that we also wanted to see whether neurofeedback success, so how well they performed in the neurofeedback, was related to these white matter changes. And whilst there was no correlation with change in FA asymmetry, there was a very strong correlation between change in the affected cortical spinal tract FA and neurofeedback success. And this was not the case in the sham group, and it also survived multiple comparison correction across 6 possible comparisons. So the participants who were better at using the neurofeedback also had the greatest increase in FA in the affected cortical spinal tract. So to summarize that all together, we found that the stroke patients were able to use the neurofeedback to increase laterality of their brain activity across runs within training days, but performance was quite variable and there wasn't any evidence of a carryover effect. Whilst we were unable to find an effect on our primary behavioral outcome measure, we did find some promising indications of effects on more gross motor function and on brain structure. For future work, including additional training of strategies that they found effective during the neurofeedback outside of the scanner may be a way to boost any behavioral effects. And also it would be useful to find ways to be able to identify and target people who are more likely to be able to use the neurofeedback. And additionally to harness the flexibility of neurofeedback in order to provide more personalized treatments. And with that, I'd like to thank you for your attention, as well as thanking all my lab members at the University of Oxford, as well as our collaborators at Maastricht University, and I'm happy to answer any questions.
Okay, so I'll be able to answer a few questions, maybe. I might not know all the details, but feel free to ask questions in the app. I have one general question that is not related to this particular presentation, but I thought might be a good question for both of us, actually, which is how do you control for motions effect in real time? Put simply, how are all the pre-processing steps done? And I guess this is slightly different from software to software, but we usually do online motion correction using registration. I don't know if you're using something, and also you can regress out the motion. I don't know if you're using that or if you're using...
So no, I only ever did, I only ever did online motion correction. I didn't regress out the motion because I think it doesn't work that well in real time. But
yes, so the most standard way of correcting for motion is just registering the data online as you are collecting it. So you're registering to the first volume, usually. Yeah. Okay, so let me see if there are questions for Zina. Okay, so there was actually a question about does one observe structural as well functional changes? Are there maybe findings from animal studies for long-term neurofeedback? as you saw in this presentation, there have been reported structural changes with neurofeedback. There's also a previous work done by Marines et al. Demonstrating that in this case, it wasn't real movement, it was imagined movement, also has structural changes in white matter. So there's been a few reports showing that neurofeedback can induce not only functional changes, like Michel has demonstrated, but also structural changes in white matter, at least. I don't recall any study looking at gray matter volume, as far as I can tell. No. Yeah. And we're talking about fMRI neurofeedback here, specifically. It's hard to moderate and read.
The app is not the best.
The app is not, yeah, there's too many. Okay, so as far as I can tell, no, I lost it. Audience response, okay.
We can also try and figure out later how to answer the questions in the app. It's
exactly.
not entirely clear, so I'm not sure we'll be able to do it, but maybe.
Yes, so there's a general question. Do you think real-time fMRI neurofeedback is scalable as a treatment in the long term? It seems resource intensive. This is a very good question. Do you want to tackle that one, Michel?
So I think there are two answers. One is, if it works really well, it's probably worth the resources. So if we can find like a large clinical change that will be meaningful, then I think bringing people in for a few fMRI sessions is actually doable. The other thing is that we could, I think if we get this to work really well, then the next step would be trying to move it to EEG or something that's more scalable. But fMRI allows us to have more control over what we're actually testing. So it's a good first step for establishing that it works, and then we can try it.
Yeah, so I agree completely. In fact, in the stroke trial, what we did is we also collected EEG measures after neurofeedback outside the scanner to try to assess if we could detect alterations in EEG that we could potentially then, pair in future studies, either with fMRI, with the idea that eventually we'd get rid of the fMRI and just use EEG, where people could use it at home, even with a portable EEG. So the idea is, right now, we're still a kind of a preclinical stage. We're trying to understand how neurofeedback works, which kind of methods and timeline is more effective. And also we have more control over the signal and also on the areas we're targeting. But potentially in the future, we could pair fMRI with EEG and have people even just use EEG signal at home. That could be potentially something. Additionally, not with the covert neurofeedback, but with the explicit kind of strategies. Could also have people just learn the strategies that work for them and just practice them at home without any neurofeedback. And that's why in this study we included a transfer trial where people are not receiving neurofeedback, but we're testing if they're able to maintain alterations in brain activity through just explicit strategies. Okay, so thank you so much for your attention and for coming, and we'll see you around. And please do send questions on the app. We'll figure out how to answer them, and you can also just email the speakers directly. Thank you so much.
Thank you.